In this screencast, we're going to talk about a couple of exceptions that we have to Mendel's principles. So, not all genes are always going to show a pattern of dominance and recessiveness. Sometimes, for two genes, there could be more than two alleles. Many times, there are traits that are controlled by more than one gene. So, let's look at some of these exceptions to Mendel's rules now. Gene expression is always the result of the interaction between both the genetic potential and the environment. So, for example, a pea plant could have the genetic capacity to be green, to flower, and to fruit, but it could never do those things if it's kept in the dark. Right? A tree is never going to grow really tall if the soil is not so good and doesn't have enough nutrients and there's no water available. So, in other words, the presence of a gene isn't the only thing that's required for the expression of a trait. The gene has to also be present with those proper environmental conditions. So the key thing to take away from this is that the phenotype, or the actual characteristics that we see, it's going to be the result of the interaction between genes and the environment. So let's start by talking about incomplete dominance, which is sometimes also called non-dominance. The most common example when talking about incomplete dominance is looking at flower color and having a red flower and a white flower. And when they combine, instead of there being one that's dominant over the other, we're going to have some mixing. And you get pink there. So all traits are not going to be as clear-cut as dominant and recessive traits. This example that I'm showing you here, this is an example we see in a particular flower called snapdragons, or another plant called 4 o'clocks. So this homozygous red, when it's crossed with a homozygous white flower, it's going to create this heterozygous pink. So let's talk about what those genotypes would actually be. This means that there's no recessive alleles, so we have to only really use capital letters here. So the red flower, we can talk about that as being R, two capital letters. And the white flowers, again, no recessive alleles, so we're going to call that big W, big W. And so the pink hybrid here is going to be a combination of what we have, so with our capital R's and our capital W's, to give us capital R, capital W. So in incomplete dominance, the genes are appearing to blend together. And again, this incomplete dominance sometimes is also called non-dominance. We don't have a dominant or recessive allele. They're blending together in the offspring. So let's talk through an example here. What if we wanted to cross two pink flowering plants? 1 and 2. What would this create for us? So let's start off. What are the genotypes of the parents? Well, I already drew that in. We know if it's pink, it's going to be R, capital W for both. So we're doing a cross here of RW with RW. Let's draw out that Punnett square. And now in this box, we'll get big R, big R. In this box, we're going to get big R, big W. In this box here, we'll get big R, big W. And in this box here, we'll get W, W. And just like we've been doing in class, let's write out the possible genotypes and the possible phenotypes. So in terms of genotypes, we have this RR. We also have a RWs. And then last but not least, we have WW. And let's write out what phenotypes correspond to that. So big R, that is going to give us red. R and W, that is going to give us pink because we have the mixing. And then, last but not least, we have WW, which will give us white. Hopefully you can see that I've written white there. And let's just count it out to make sure that we have the correct ratios here. We're going to have 1/4 be big R, 2 out of the 4 are RW, or pink, and 1 out of the 4 are WW, or white. So when you're talking about what it's able to produce, you can talk about it in a fraction, or you can talk about it in percent, and you could say 50% of the offspring will be pink, 25% will be red, 25% will be white. But in certain plants, we could say that the flower color is showing non-dominance, incomplete dominance, but the stem length is showing dominance, right? So the allele for the long stem is dominant over the allele for the short stem, and we could do a cross here of a heterozygous long-stemmed red plant, crossing that with a short-stemmed pink plant. So spend a second here thinking about what are we going to have for the genotype of the first parent, and then what will the genotype of the second parent be. And let's use L's as the alleles for long versus short, and for pink versus red and things like that, we will continue to use R and W. So the genotype of the first parent, hopefully you figured out, would be big L, little l, since it's heterozygous, and we know that it's red, so it can only have R's. And then the second parent, if it's short, we know that short is recessive, so we're going to be little l, and pink we know is a combination of red and white. So now we need to add these alleles in, either up here or on the side, and then add these alleles in, up here or on the side. But before we can do that, we need to figure out what all of the possible combinations are here of the alleles. So remember, this is when you do your distribution, so you can't just put big L here, little l, R, R. That doesn't work. You have to distribute them out first. So big L can go with big R, big L can go with big R with big R, little l with big R. So these four are going to be what we put in here. And now let's distribute for here. We need little l can go with big R, little L can go with W, little L can go with big R, little L can go with big W, which gives us little l, big R, little l, big W, little l, big R, little l, big W. So again, what we have going on here will go up in these boxes, and what's down here will go on the left side of the Punnett square. So let me draw those in now. And if you're watching this at home, take a second to pause and fill in your Punnett square chart, and then check with mine once you've done it to see if you figured it out right. So pause now, fill it in. So this is what I got. Now let's go through the genotypes and phenotypes. We know there are 16 boxes, so we'll find our ratios here. But we have one genotype that is big L, big R, one that is big L, big R, big W, one that is little l, R, little l, R, W. So those are we only have 4 genotypes there. And in terms of phenotypes that it gives us, this first one will give us a flower that is long-stemmed and red. This one will give us one that is long and pink. The third one will give us a short and red. And the fourth one will give us short and pink. Nope, and now we can't see everything on the slide, but back to genotype, phenotype. Hopefully you can see all of that. So there we go. That's talking about incomplete dominance and working through 2 examples for you. Next up is codominance. And a really good example to use when talking about codominance is when talking about blood type. So there actually are 3 alleles that we use when we're determining blood type, and we write these alleles as I have written here. I with a little a above it, an I with a little b above it, or just a little I. Those are our 3 possible alleles. And the allele IA and IB are going to be codominant, and the little I will be recessive. So codominance is when both dominant alleles are going to be apparent in the phenotype of the heterozygous offspring. Let's talk through all of the possible genotypes for blood types. You could have an individual that is capital I A, capital I A, and so they have both of the alleles for type A blood, so their phenotype is type A blood. You can have an individual that is capital I A with a little I, but we know that the little I is recessive, so that individual will still have type A blood. You can have an individual with big IB, both alleles for type B blood. So their phenotype would be type B blood. You can have an individual with the capital IB allele and the recessive little I allele. That will still give you type B blood. Then you can have a mixture of capital IA, capital IB, and that individual will have type AB blood. And since both of those alleles are codominant, both are going to be expressed in the offspring. And then last but not least, you could have an individual that's little I, which is what we call type O blood. So make sure you're comfortable with all of those genotypes and how they're expressed as phenotypes in blood. And let's go through an example here and figure out what types of offspring might we expect if we have one parent that has type AB blood and the other parent is going to be heterozygous for type A blood. So genotype of the first parent here, we're going to draw out their alleles, which will be capital IA, capital IB, and the second parent, because they're heterozygous, we know that they're going to be capital IA, and instead of having another capital IA, because they're heterozygous, they're going to have the recessive little I. So go ahead and do out a Punnett square of these 2 parents. These alleles can go in up here. Let me do a different color. IB, and then these alleles will go over here. IA, little I. Let's pause for a second and draw in what you think will be the resulting genotypes for the offspring. So this is what I got. Let's write out the possible genotypes and phenotypes. We have individuals that have capital IA, capital IA, capital IA, IB, capital IA, little I, and capital IB, little I. And what do these all make? This is going to be type A blood, this will be type AB blood, this will be type A blood, and this will be type B blood. And the ratios here are going to be 1/4, 1/4, 1/4, 1/4. But if I were to ask you what percent of their offspring will have type A blood, you'll have to remember to combine this genotype and this genotype. So individuals with type A blood, there would be 2 out of the 4. 1 out of the 4 would have type AB blood, and 1 out of the 4 would have type B blood. Let's just walk through another example with blood type here. Let's say that we have a man and a woman, and they're having 4 children. Each of their children has a different blood type. So what are the genotypes of the parents and the 4 children? Pause for a second and see if you can figure out the answer here on your own. I'll draw it out. In this situation, it sometimes is good to draw it out and just double check what you think the parents would have and see if you actually create 4 different blood types. But we know that, looking back, we have only 3 alleles, so we need to create as many combinations of these as possible. So the parents are going to have the alleles. One will be capital I, A, little I, and another parent will be capital I, B, little I, right? Because then we could get blood type A, blood type B, blood type AB, and blood type O by having the A child be IA little I. The child with type B blood could be capital IB little I. The offspring with AB blood type would be big IA, big IB, and the child with type O blood would be little I, little I. Unfortunately for us, blood type isn't just type A, type B, type O, and type AB. There's another component to our blood type, which is called the Rh factor. So you could have people that are either going to be Rh positive or Rh negative in their blood. So the Rh factor will be determined by one gene with 2 alleles. And the allele for Rh positive is going to be dominant over the allele for Rh negative. So we're going to use capital R to designate the positive allele and little r to represent the negative allele. So let's try out a practice problem here. Let's say a woman who is blood type AB negative. Let me go to a new slide here. AB negative. She marries a man with type O positive blood. And the man's mother had a blood type that was A negative. So let's talk through the genotypes first. So let's talk first about this woman here, who's AB negative. So let's first do her alleles for the blood type. So to have AB blood, she'd have to be capital IA, capital IB. And then she is negative, which is recessive, so she'll have to be little r, little r. The genotype of the man, for type O blood, he has to be little I. And then we know that he's positive, so he can either be big R, little r, or big R, big R. But let's look at his mother, and we know that she was A negative, so her genotype for blood type will have to be capital IA and either another IA, or she could be capital IA little I. And because her offspring has a little I, we know that this is the one that she is. And because she is negative for her blood type, she'll be little r, little r. And if the mother has the little r present, then her son must have received it. So he has to be big R, little r. So I know it's kind of complicating adding the sort of mother in there to figure out what the son's genotype is. That's a little more complex than I'll ever ask you on an exam. But let's work through here. and see what happens when we cross our female and male. But first we need to distribute out and find out what their alleles are, their allele possibilities are. The woman, she can be IA little r. She can be IA little r again. She can be IB little r. And she can be IB little r. So we have to use these here and put those into a Punnett square. So your Punnett square should be set up like this. Go ahead and pause and then write in all of the genotypes that you find for their offspring, and then you can check it over with what I find. So this may look confusing to you since in the boxes now we have 1, 2, 3, 4, 5 letters, but keep in mind that this IA, you count that as one allele. So go ahead and pause now and write in for me what you get for your possible genotypes and phenotypes. Write those in, figure them out, and see if you get the same ratio as me. So I got 4/16 for each of those genotypes, and so we know that capital IA little I will code for type A blood, and the big R little r, because big R is dominant, will give us a positive. It's dominant, so meaning that positive is the dominant trait over Rh negative. Down here, capital IA little I will again be type A blood, but the little r will give us negative. Then down here, capital IB little I, that will give us type B blood, and the capital R little r will give us positive. Capital IB little I will give us type B blood, but the little r will give us negative. So there we go for our ratios. Hopefully you were able to follow along there. Again, this is the most complex that you can get, and I wouldn't ask something like this of you on the exam, but just trying to show you how complex it can get when you have multiple factors playing a role. And sometimes you have dominance and sometimes you don't, so you have that mixing of codominance or incomplete dominance with regular dominance or recessiveness. Okay, that's the end of this screencast here on codominance and incomplete dominance.